Biology and of physics at Harvard University, and he is one of our investigators in the mathematical modeling of living systems program that we do here at the foundation. And his work, exactly in line with the goals of the program, is he combines theory and experiment to bring quantitative methodology to the field of life sciences, in this particular case, evolutionary dynamics. And he's going to tell us about evolution when selection is pervasive. Michael?
All right, thanks, Andy. So what I'm interested in primarily is how the evolutionary process works. Basically, what we're trying to work out is how these sort of basic forces of mutations, natural selection, genetic drift, these kinds of things that Darwin could have told us about, work together and in some sort of probabilistic sense filter from some set of things that could in principle happen in any given natural population to determine what sorts of outcomes we see in evolution and what kinds of patterns of genetic diversity this process leaves as signatures within a population. And so, as Andy just said, my work, we use a combination of experimental evolution, primarily in budding yeast, as well as theoretical population genetics to try to address these questions. And this is obviously a very broad audience, and so what I'm going to try to do today is try to give you guys a sense, at least a little bit of a sense, of what the main problems are. And then I'll talk quite a bit about experiments to hopefully give you guys a sense of what kinds of data we can collect, and hopefully this will make the problems a little bit more clear. And I will sort of give a brief overview of the kinds of theoretical approaches that we've taken. So one of the most basic things that we know about evolution, of course, is that if you take some kind of population and you expose it to some kind of new selection pressure, the population will typically adapt in response. And there are many, many natural examples of this that have been characterized in great detail. So here are a couple of particularly well-known examples. So these are mice studied by Hopi Hoekstra's group down the hall from me. And you can see that there are various different coat colors of these mice. And basically what happens is if you take a brown mouse and you put it in a habitat dominated by this lighter colored sand, you'll get adaptation of the mice to match the color of the sand and vice versa. And so it's not hard to imagine why these kinds of traits might be adaptive. Here's the example of Darwin's finches. These are a bunch of different finch species that live on the Galapagos. And if you look closely, you'll see that they all have different beak morphologies. And those beak morphologies are generally optimized in order for for that particular lifestyle of that finch, what kind of diet it primarily eats and where specifically it lives. And through really a lot of tremendously painstaking work in evolutionary genetics, we can now really say quite a lot about these kinds of processes. So you can say what specific mutations underlie these changes in coat color or are responsible for these differences in the beak morphology of these finches. And we can say, when they occurred, what kinds of advantages that they have provided. So all of this work, though, is primarily sort of observational and largely descriptive. The thing that it's really difficult to do in these kinds of natural settings is say anything about why evolution has acted in this particular way as opposed to some other way it might hypothetically have acted. In other words, if we were to somehow repeat this evolutionary experiment, what would we expect to see? Would we expect to see the same phenotypic outcomes? If so, would they have the same genetic basis? And inevitably, of course, the answer to this kind of question is that in some circumstances you would see the same outcomes, other circumstances you'd see different things. And the key question we want to get at is what determines the answer to this question? Why do you see more similar or more different responses? What aspects of evolution are more deterministic and what are completely random? So, as I said, we look at this question using a combination of theoretical and experimental approaches. I'm going to tell you a lot about laboratory evolution experiments, which I think offer a really great opportunity to just watch what's going on in evolution and start to get a handle on these kinds of questions. So basically, the upshot of all these evolution experiments is you take some typically microbial population and you let it evolve in the lab in some random selective pressure that you impose in the laboratory. So this is the most famous example of experimental evolution. This is a picture of lines of E. coli maintained by Rich Lenski's group. They've been evolving now for about 27 years in his lab, corresponding to more than 60,000 generations. There are a dozen lines, and I'll tell you about some observations that we have recently made about evolution in these populations. We can also, in my lab, we primarily do evolution in yeast and in thousands of replicates, but the basic idea is the same. And it's a little bit less pretty than the previous pictures, but we can see phenotypic changes in these populations over time. So if you look through 50,000 generations of evolution in these lines, for instance, the average fitness of these populations is increasing. And here, fitness, I'm going to talk a lot about. Fitness here depends on lots and lots of different things, but from an operational point of view, the fitness of one of these E. coli strains is simply its tendency to leave more or less offspring into each successive generation. And so this is a precisely measurable quantity in these experiments. You can take evolved strains of E. coli, you can resurrect their ancestors from the freezer, and you can compete them, and you can look at how the relative frequency of these strains changes with time, and the rate of change of those frequencies is what we're measuring here by fitness. So the more evolved lines will outcompete the ancestral lines. So they increased in fitness. And then we can very easily nowadays figure out the genetic basis of these changes. So as I mentioned, you can store these populations in the freezer, and they've been doing this since the 80s, and now sequencing technology has gotten much more advanced. And so this is a figure now completely outdated just from a half a dozen years ago, where Rich's group took a single clone from one of these lines, basically a single E. coli cell, and sequenced it at 5 different time points from generation 2000 to 20,000. And what you're seeing here is each of these circles is one of those clones from the early time point in the middle to the later time point at the outside, representing the genome as a circle, 4.5 megabases. And there are a bunch of tick marks here. Each of these tick marks represents a mutation that occurred, that was found in that clone. And you can see how the mutations are accumulating in time. And these sort of cryptic things, how many of you know E. coli? Anything about E. coli? All right, a few of you, all right. Those of you who know something about E. coli can probably tell various kinds of stories about why these things might be beneficial. Mutations in genes like PYKF and SPO-T and so on. I'll talk about. But regardless of this, okay, we have some kind of pattern of the accumulation of mutations within this population. And the great thing about this experiment, as opposed to some sort of natural population, is that we can now start to directly ask questions like, what happens in an identical replicate population? How many of these mutations would we see again if we sequence another replicate population? What fraction of the populations will have specific mutations and why? And essentially, can we sort of start to tease apart how these various evolutionary forces determine the outcomes? And I'll just sort of, I'll give you a hint of the answer. Actually, I'll tell you the answer. The answer to that question is no. We can't really predict any of this stuff yet, but that's the sort of thing that we're trying to work on. Okay? Yeah, please interrupt as much as you like, yeah. Yes, in a probabilistic sense. We want to say, for example, this mutation, what fraction of the time will it show up in a population, right? And yeah, so we're talking about prediction in a probabilistic sense.
Yeah, the question is just the practical thing. How long does it, how fast, what's the time scale?
I'll tell you more about this, but this is 60,000 generations has so far taken about 27 years. It's about 6.6 generations a day. You could do more. In my lab, we typically do yeast at roughly 10 generations a day. The generation time of these E. coli is like roughly 20 minutes. You could do much more, but then you would have to come in the lab every 4 hours or something. So, over 27 years, that would get tiresome. Yeah. Was there another question? Yeah. Yes, so they're started with exactly identical genotype, evolving in exactly identical conditions, at least as best as we can control it experimentally. Yeah.
So you said you can freeze these. Can you just put them on hold, put them in the freezer,
Yep.
take them out 10 years later? Wow.
Yes, so it's awesome, right? So you just freeze them down at minus 80 degrees and 20% glycerol, and you can resurrect them 50 years later if you like, and you can just grow them again just fine. All right, and so it's been really cool that a lot of things which were completely like nobody even thought of the ability to sequence these things when he started this experiment, but because you keep them frozen, you can just do all this stuff. All right, so basically what we're trying to get at, right? Like all of these kinds of data that we're collecting are a signature of this evolutionary process. We know these basic rules, right? Darwin could have told us these basic rules, but we want to understand how they act together. And basically, like what we're doing is there is some set of things that could provide advantages in this environment. Many of these mutations that you observe and many others that you don't observe in this population just due to chance. And somehow, they have some rates, they have some fitness effects, and somehow this process is picking and choosing and determining what we see. So, why is this complicated? All right, so the basic problem is that genomes are like physical strings. Right? And I'll describe what I mean by that in more detail. So for a long time, people thought that the primary sort of limiting factor in adaptation is waiting for mutations to happen. Right? The idea is that beneficial mutations are rare. The main limiting factor is waiting for the damn things to happen in the first place. Okay? And so that led to this kind of picture, which I'm drawing schematically here. Time running on this axis, and we imagine lining up the individuals in the population here. Right? And so this is known as a Muller diagram. So the idea here is that at that early time point, which I can't reach with my pointer, some mutation happens in some individual. Okay? It happens in a single individual, so genetic drift is going to be important while it's rare. There'll be fluctuations, but provided it survives those, if it's beneficial, it'll quickly spread through the whole population, and everyone will have mutation A. And we say this mutation has fixed. All right? And this process is known as a selective sweep. And then sometime much later, maybe some other beneficial mutation B will occur, and there'll be another selective sweep, and so on. And the process is just determined by this sort of sequence of selective sweeps. And we can think of mutations are occurring as a Poisson process. They surviving drift at a rate which is given by their selective effect. And I mean, we can very easily say how you're going to pick and choose among the possible outcomes. And this kind of picture has underlined most of our methods of analyzing sequence data. So you could imagine you take a bunch of individuals from a population and you sequence their genomes. So each individual is a line here. Their genome is sort of schematically represented. Maybe you have some neutral mutations represented by these open circles, but the idea is that only one selected mutation at a time. So if we get some mutation that is going to be positively selected, the idea is we can just focus on its dynamics independent of everything else that's going on. And so there are models from the 50s and earlier even that really have completely understood this process. It's just some simple stochastic differential equation describing genetic drift and selection and so on. And if the mutation does well, it will increase in frequency. This is sort of on the way to fixation during a selective sweep. And so that's relatively simple. Okay. But of course, if you think about how inheritance works, right, you do not pass on individual loci to your kids, right? If you have kids, you're going to pass on whole chromosomes, right? And if you're lucky enough to have grandkids, your kids are going to pass on to your grandkids big chunks of your chromosomes, like hundreds of megabase chunks of your chromosomes. You can't just get, one thing but not another. So, many of you may have deleterious mutations somewhere in your genomes. Maybe some of you are lucky enough to have beneficial mutations. You are going to pass them all on or neither, right? So you can't pass on things independently. And so, in fact, what we see happens, and I think over the past 10 or 15 years, it's become clear that this sort of picture is actually everywhere, certainly in microbial and viral populations, as well as our own evolution. Natural selection is typically not rare. And so instead we have this situation, lots of different mutations. Here I'm drawing a half a dozen, or maybe 5, I guess, beneficial mutations occurring in different genetic backgrounds, different individuals, and they all start to spread. And because they have to be inherited together or not at all, what happens is these things can't, unless there happens to be a very lucky recombination event, they can't combine. Instead, they have to interfere with one another. And there's a whole sort of traffic problem, which is preventing them all from fixing. All right, this problem generally is known as clonal interference, a word I'll use more as we go along. But all this traffic leads to this whole swarm of different things going on and competing and interfering. And it's much more complicated in this kind of setting to understand how evolution is going to act. Because basically what you have is when you have these genomes now, okay, if these mutations now, there are many mutations that are selected, whenever we get some new mutation occurring, it depends, the fate of this mutation depends a lot on all the other stuff that's going on. And so for instance, natural selection can't increase the frequency of this beneficial mutation without also increasing this deleterious mutation, unless you happen to get a recombination event right in between, which on local scales in the genome is quite unlikely. And so basically this problem makes natural selection much, much less efficient. All right? Yes? So basically anything can exchange genes at some level. So if I have time, which I probably won't, I'll talk about our yeast experiments where we vary the recombination rate. We look at purely asexual populations and we look at different recombination rates and look at it. But basically, what I'm going to describe to begin with is thinking about evolution within a sort of chunk of the genome that is effectively not recombining over the relevant time scale. And if we want to think about the evolution of genomes of obligately sexual organisms such as ourselves, for instance, then we need to think about what that distance scale is. There are distance scales within our genomes that evolve basically asexually over the relevant time scales, and then different chromosomes more or less evolve independently. And that's a sort of open question exactly how we should think about that. But what I'm going to do, at least to start, is talk about evolution within a chunk that we can think of as being inherited together for the moment. And so in the E. coli populations I'll talk about, there's no recombination. These are just, they're short genomes, right? 4.5 megabases, no recombination during the experiment. Yeah.
Can you just, I just want to make sure I'm following relative to the simpler picture and the previous lecture. So the idea is that here, in reality, you don't sweep through the whole population. The mutation doesn't sweep through the whole population before a new mutation takes place.
That's right. That's right. So there's lots of things happening. You can no longer think of selection acting on individual mutations in a sort of temporally separated way. And so what that means is that you can't just pick out all the good mutations and get rid of the bad mutations. And sometimes you get good mutations on bad backgrounds and they go extinct and things like this. Okay. So we have to think about the whole genome simultaneously. Yeah.
Did I understand correctly that whether the extent to which a mutation is beneficial or not is also independent among different mutations, not that
yeah.
One mutation is beneficial only if another mutation is also.
Right, so this is a major topic of research in my lab and in many other labs. So the phenomenon you're describing is known as epistasis. So I'm going to talk about mutations just because I don't have time to talk about everything. Today, I'm going to primarily talk about mutations as if they have some fitness effect that's independent of the genetic background. Of course, in general, that's not true. Often it can be the case that a mutation happens and its fitness effect depends on the genetic background in which it occurs. In general, in these kinds of experiments, that's like a second order, that happens a lot, but it's not the generic behavior. But there's a lot of really interesting questions to be addressed with that. And obviously that's going to make evolution at some level less repeatable because you have to get one thing to get another. But yeah, that's for another day maybe. Thanks. Okay, so all right. I think I've probably sort of summarized the sorts of questions we want to address. But basically, just to emphasize, what do we want to do? We want to sort of understand how efficiently evolution sort of picks out the best solutions. What is the probability a mutation will fix? How similar or different are mutations in replicate lines? As we'll talk about in a little bit, we can also imagine sequencing populations from a snapshot in time, right? This is usually often what we're limited to. Like for instance, if we sequence human populations, we can't freeze humans at -80 yet. So we're limited to basically today. And so we could sequence all the people in the room, okay? And this is the kind of thing now, this sort of scale of experiment is something that I could do in my lab. I won't, but we could sequence this room. That's a sort of a scale of something that we can do. And then we could imagine looking at the patterns of genetic diversity, basically the similarities and differences across our genome, and there are various ways we could summarize those statistics. And we want to try to predict what we expect to see in this kind of data. So, all right, so hopefully this gives you a sense of the kind of the problem, all right? And so before I talk, I'm not really going to talk about any solutions, but I'll try to give you a sense of the solutions. But before I do that, I just want to sort of show you some data, okay? And this is data, I'm just going to show you, this is the latest data that we have from my lab, where we've collaborated with Rich Lenski, who conducted this experiment that I've told you about in E. coli. And basically the upshot of this experiment, as I've already kind of mentioned, is you put some E. coli in a flask. The next day you come back and you put it in and put a little small aliquot, as it's called, in a new flask, and you just do this for 27 years now. So a population has been it fluctuates on very short time scales in population size, but basically this amounts to what we call an effective population size of about 30 million or so, all right? And as I said, this has now gone 63,000 generations as of now. And they have frozen down samples every 500 generations, okay? And there are 12 replicate lines, all right? And so, all right. Just to give you a sense, this is a long time compared to even, things in human evolution, all right? And so the first thing that you can see is how the fitness has changed in these populations over time. All right, and so each of these lines is one of the independent replicate populations, and the black line is the average. These measurements kind of stink for various technical reasons. We now have much better ways of doing this, but this experiment was founded before they were available, so we're stuck with this. So anyway, these have been conducted now through the first 50,000 generations, and there's a new paper now, just in the past few days, out to 60,000 generations. And what you see is that these populations have all increased in fitness, primarily in the first few thousand generations, okay? They do, however, continue to increase in fitness this whole rest of the time. You can compete the 50,000 guy against the 45,000 guy against the 40,000 guy. The later guys are measurably more fit than their ancestors, which, frankly, I find kind of astonishing. They're continuing to get fit in the exact same environment for 60,000 generations now. Yeah.
So is this environment that they're in, is this particularly different from the original environment they were in at time zero? Or
No,
when you brought them
it's the
in?
same environment. It's exactly the same thing.
Why did E. coli evolve to be fit beforehand? Or were they just not evolved to live in flasks?
Well, after 27 years, one would think they would have got used to it, but, they haven't.
What was happening is at time 0 minus epsilon.
Yes.
What was different? Why does the fitness rapidly increase after you put it in the flask?
Well, so, I mean, supposedly the E. coli were adapted to this laboratory environment. Personally, okay, we don't really know, but my guess is that actually laboratory yeast are primarily adapted to going into and coming out of freezers, and E. coli too, right? Anyway, there's various things that they were doing in standard laboratory maintenance. You also passage through single cell bottlenecks typically in the lab, which can cause the fixation of deleterious mutations. For whatever reason, they were kind of lousy at the beginning, okay? And they quickly got better, okay? And then they continue on. And, in my lab, whatever, I'm not old enough to have done this experiment yet, but we do, thousands of populations of yeast for shorter periods, a few thousand generations at most, and we always see the same kind of thing, right? Yeah.
Fitness, is it a transitive property?
Yeah, so, there's another long story here. Effectively, in this experiment, it is mostly a transitive property, and people have checked that, but it is not always a transitive property. And as we'll see, there are also sort of ecological interactions that get created that cause frequency-dependent fitness interactions. And I'll talk about some of those. But so loosely speaking, it's usually more or less transitive and more or less frequency independent for most things, but not always. So there's other complications that we could talk about there, but I won't. Yeah. Well, I mean, presumably they cannot divide every 30 seconds. So at some point there has to be an upper bound. But 60,000 generations, they haven't found it yet. Okay. So my lab and various others has done a whole bunch of sort of analysis via sequencing of various other kinds of experimental systems. So this was our work here primarily with thousands of populations of yeast. But basically, these experiments all sort of showed this pattern of rapid adaptation, clonal interference, and so on, all right? But they were all sort of limited, just to sort of put these to scale, to this first bit of the experiment that we have access to here. And so basically what we wanted to do was look at what's going on. Yeah. The way it's done is basically you have a reference strain, okay? And in practice, we make lots of different reference strains. But you have a reference strain which has some kind of marker. The reason these fitness assays are kind of lousy is that marker is essentially a drug resistance. Now we have various fluorescent markers that are better. But the point is you have a marked strain. You mix it in exactly the condition of the experiment with your evolved strain. You measure the relative frequency. You maintain the population according to exactly the protocol of the experiment, and then you measure the frequency again. And the change in frequency is telling you, which strain is winning and how quickly, and that's the fitness. Okay? And somebody asked a moment ago about transitivity, and so one thing you can ask is if A is bit fitter than B and B is fitter than C, is A fitter than C, and right? And it turns out for most of what I'm saying, this is all perfectly transitive, but not, there are exceptions, of course, but yeah. Well, it depends on what you change. I mean, these are kinds of experiments we are doing, and other people are doing all these kinds of experiments, but it depends on what you change it to. Sometimes, yes, right? Presumably, can be complicated structures of trade-offs between environments, but on the other hand, adaptation to other pairs of environments might be highly correlated. So the answer is it depends, as is often the case. All right, so let me just tell you about the kind of data that I'm going to show you. So what we did was Rich sent us all these, things from his freezer, and we took whole population samples from not quite, but almost every time point through here. All right? And the idea is what we do is we take this sample, we grind up the whole population, and we sequence it. And the idea is if there's some mutation, let's say it's at 30% frequency, that mutation at that time point, you should see 30% of your sequencing reads support the mutation, 70% support the original nucleotide there, and you infer, okay, that was a mutation at 30%. Obviously there is a whole complicated bioinformatics problem here because you are trying to push down, this is a lot of sequencing, you're trying to push down coverage, there are sequencing errors, etc. So there are interesting computational problems here which I won't talk about, but suffice it to say that in the kind of method that we developed, we are going to be able to see any mutation that reaches a frequency of at least, say, 5 to 10% at some time point, and then track its frequency through time. And I just want to emphasize, right, so this is already, we are only looking at very successful mutations, because the population size is like 30 million, so there are like 5 orders of magnitude below what we can detect, right? Maybe even 6 orders of magnitude below what we can detect, all right? And there's lots of interesting dynamics going on there, and one of the sort of interesting technical things that we're trying to build is how can we see all of that, all right? But it's a complicated problem. And so today what I'm going to tell you about is this. So I just want to emphasize these are the sort of successful mutations. But even given that they reach this 5, 10%, there's all kinds of interesting things that we can see about what happens to them. All right? So let me just show you. This is just to sort of prime you for the kind of data we're about to see. All right, we're going to see time courses like this. On this axis is the frequency of these alleles, and we'll see these lines. And so what happens early on, right, in the first 500 generations, we see some new mutations arise, and this is how their frequencies have changed over the first 500 generations. Okay, they're de novo mutations that occurred spontaneously in this line. And then we watch what happens to them. So here, these mutations fix. This one goes slower. These ones went extinct. All right, and we see this process now. New mutations arose and started to fix. Other ones went extinct. And there's this whole complicated process that we can just sort of play out. Okay? All right. So there are 12 lines. The limiting factor actually in doing this is getting them out of the freezers. So far, we've done 4 of the lines, and this is what they look like. Okay? So each of these panels is an independent population, started from an identical genotype, evolved forward in time. Each of these lines within a panel is a mutation, and its frequency is a function of time. And of course, we know what these mutations are, and I'll talk in a few minutes. You know, many of the same genes are mutated in replicate lines and so on, and we could tell stories about that. But the first of all, sort of just gross level thing is that, you know, there is clearly a ton of stuff happening. And this is the kind of data and process that we want to understand, right? Why, you know, there's something sort of the same about all these lines, but there's lots and lots of differences as well. And we want to understand, you know, what can we understand about these kind of dynamics? Yeah. It's just there are so many lines here that, is this what you're talking about? There are thousands of mutations here. All right, I'll spoil a little bit of what's coming. One of these mutations is a mutation in a gene called MUT-T. It's involved in the mismatch repair pathways in E. coli, and so when this mutation fixes, it elevates the mutation rate going forward. Okay, and I'll talk about that more in a second. Yeah.
Usually, I see many lines going from 0 up to 1.
Yes.
I have not yet found any going from 1 down to 0.
Yes. Once you fix, you're fixed, right?
Okay.
Because now everyone has you, and so all the descendants of that will still have that, unless you get a precise back mutation at that point. Yeah. Okay. All right. So, all right. This population is really cool for various reasons. But I'm going to talk about analysis of these three because we just got this one recently, and so we just haven't done the analysis yet. So first thing we see, this is the fitness of these three lines that I'm going to talk about here, all right? And, you know, it's just from that previous plot, looks basically the same. You see the fitness increased quickly and then slowed down, but it's still increasing. Now, if you look at the molecular level, okay, if we look at all the sort of number of mutations that have fixed as a function of time, okay, you see this steady increase, right? It does slow down a little bit, okay, but not much at all, not nearly as much as that slows down. And of course, we have this one example. This is that mutator that fixed and jumps up now, you know, orders of magnitude higher than in terms of the number of mutations. But one interesting thing is, right, okay, you can obviously see which is the mutator population from this graph, all right, but you can't tell from this graph, I would argue. So it's kind of, right, this must have been a selected event, right? Elevating the mutation rate was presumably beneficial here. Yet, I mean, there's not that much of a signature of it, right? Okay. We can say a lot. Of course, we know what all these mutations are. And so, you know, one thing you can start to do is classify these mutations into different genes and so on. I'm not going to belabor this, but suffice it to say that these mutations are overwhelmingly functional, right? So mutations in coding regions, for example, can be synonymous or non-synonymous. So synonymous mutations don't change what the protein codes for and are presumably usually at least mostly non-selected. Non-synonymous mutations do change the amino acid. So just by chance, you would expect, you know, roughly 1/4 to 1/3 of all mutations in coding regions would be synonymous, and in fact, you see much less than that, okay, indicating that these dynamics are driven by selection, which I guess we knew anyway. We also see a lot of structural variation, primarily insertion sequence mediated events. And I just want to emphasize, right, this is all driven by selection, right? This is all this kind of swarm of events that I was talking about earlier. If there were no selection, right, even though this is a very long time scale of an experiment, we would expect genetic drift to be able to change the frequencies of these alleles by less than about 1% over the entire course of the experiment, based on what we know about genetic drift. So this is really dominated by selection and all these hitchhiking events, all right? So obviously, clonal interference is widespread. Just to zoom in on the kind of things we see a lot of, okay? So I'm just zooming in on this little time frame of this population just to illustrate the kind of thing you see a lot, all right? And here are just a few of the mutations, just to highlight the basic things. So here you see this mutation starting to increase in frequency, all right? This group of additional mutations arises in a competing background and starts to increase, and it looks like it's going to outcompete this group. Then a further mutation happens to occur in that genetic background and this one winds up fixing and this one winds up going extinct, right? These are all beneficial mutations, or at least most of them, all right? But nevertheless, right, this sort of complicated dynamics of which thing happens on which background is determining what you see, okay? I'm going to skip that. All right. So we see more signatures of these sort of effects of interference if you look at the specific genes. So here's just a couple of examples, all right? These are 5 different mutations in the gene PYKF, which appear in this population, okay? These are all, they all reach 5–10% at some point, right? So these are already exceptionally successful mutations. We know they're beneficial, actually, from lots of independent work. So selection is trying to drive these to fixation, and it succeeds in 1 or 2 cases, and others wind up going extinct, okay? These are just distinct mutations in the same gene. We see a major excess of this parallelism above what you would expect by chance. Again, that's not surprising, indicating these are things that, that natural selection is actively picking out. But here's another example of a mutation in this gene SPO-T. We see also 5 examples of this that reach detectable frequency in this population, but they all go extinct. And this is despite the fact this mutation fixed in one of the other replicate populations. And one of the other cool things, which I haven't mentioned before, is in these kinds of systems, you can actually recreate these mutations. So we actually, well, we didn't, Rich and other folks, took these 2 mutations, okay, these 2 genes, and made several of these mutations in a bunch of different genetic backgrounds, and found that they are essentially beneficial in any genetic background in which they occur. Actually, this mutation appears to be more strongly beneficial than that one. But what you can see here is that the dynamics of this process are as important as biology in determining what happens. All right. So, as I mentioned, we do a bunch of theoretical work to try to understand this process. But I'm not going to belabor all of the details. But I want to give you guys at least a sense of how we think about this. So the basic idea is that we cannot write down, or we can write down, but we can't make any progress if we're trying to sort of track the frequency of every possible genotype that could arise in the population. It's completely an intractable problem, at least as far as anyone knows. But instead, what we've been trying to do is recast these as sort of front propagation problems, all right? And basically, the idea goes like this. Okay, there's a lot of stuff happening in the population, right? There's all of these mutations that are occurring, and they are generating some variation in fitness within the population, right? This is just sort of collectively all the stuff, and I'm just drawing it schematically with this blue figure here, right? This blue distribution. Obviously, what this distribution is a complicated problem, and of course it fluctuates all over the place, and there's interesting things to work out about that. But on average, we get some fitness distribution, and we can characterize its fluctuations to some extent. And then we think about what happens to any individual mutation in terms of its interactions with this rest of the variation. So here what I'm drawing an example of is a mutation that has some modest benefit, okay, this much of a benefit, that happens to occur in a genetic background that's somewhat more fit than average. And it creates this lineage that's substantially more fit than average. And so then what's going to happen is this guy is more fit than the average dude, so it's going to increase in frequency, right? You're going to have more offspring than everybody else, okay? And so it's sort of increasing in frequency. And in the meantime, all this other stuff is still happening and the rest of the population is catching up, right? And if nothing else happens in that lineage, what's going to happen is eventually the rest of the population is going to pass it by and it's going to go extinct, right? Or alternatively, what could happen is that additional mutations could happen in this background and it could sort of surf ahead of the rest of the population, and that's how it's going to fix. Right? And so we want to understand this kind of surfing process. And so there's theory that we've been doing to try to do that. The basic sort of qualitative picture is that, of course, the only thing that matters in terms of how well you surf is how far out ahead you got here, right? The sort of further out ahead you got, the bigger head start you have, the more likely you are to eventually fix.
Why is the rest of the distribution?
Because there's selection is killing these guys and pushing these guys and favoring these guys, and other mutations are happening in other genetic backgrounds. And we can think of this as sort of creating collectively some average increase in the rest of the population, right?
I would have thought those little guys, that little red one,
Mhm.
is the dynamics of the average.
No, this is the dynamics of a specific lineage, and we're coupling it to the average dynamics of everything else. And so the average dynamics of everything else, there's lots of stuff going on, but on net, that's making the population adapt. And then you're sort of comparing this lineage to everything else, right? And the upshot is basically this creates this sort of tension, right? There are big effect mutations, things like this PYKF and SPO-T and so on. These are sort of things that you see again and again in replicate populations. They are presumably the sort of best thing that you can do, all right? But by definition, right, the best possible solutions are rare. And so, these things can sort of jump out far ahead and have a good chance of fixing, even if they occur in sort of generic genetic backgrounds, right, because they provide a big advantage. But they don't happen that often. And on the other hand, there are lots and lots of neutral, deleterious, modestly beneficial things. There are many of those mutations happening, but they can only succeed if they happen in these exceptionally lucky backgrounds. So there's this kind of tension between how common are these large effect things versus how common are these high fitness backgrounds. And that determines sort of how efficiently selection is picking out this sort of good stuff versus lucky stuff that happens in good backgrounds, right? And so we can try to quantify that, all right? So that's basically how this theory works, all right? So I just want to point out a couple of complications, right? So somebody mentioned, a few people have asked about whether fitness is transitive or not frequency dependent. And there's some really sort of cool things that we observe in these experiments that indicate deviations from this sort of simple perspective that I've told you about so far. So here, for example, this is this population that I have shown a bunch of times. Here I'm just highlighting a couple of mutations, okay, to look at their dynamics, okay. So if we look at this mutation in top A, it establishes within the first 1,000 generations, appears to fix very quickly. But in fact, it is not totally fixed, because we can see much later on, it and many other mutations that sort of travel along with it decline and then come back up again. And the opposite happens to this gene SPO-T, okay? And so what must be the case, right? This mutation in top A is not quite fixed. This mutation in SPO-T is not quite extinct. They're maintained presumably by some stabilizing fitness interaction. Maybe they're cross-feeding each other, or who knows. And then later there's various events that alter this frequency in various ways. And you can see some of these things. So for instance, this is this top A mutation here, zooming in on a bit of it. You can see some mutations sort of sweep within that group, and then other mutations occur later and drive this change in the relative frequencies of these types. Okay, and this plays out over a very long time. Here's another example, which is really particularly cool. So if we look at a couple of the mutations from within this population, this is that MUT-T mutation, which we talked about earlier, that increases the mutation rate. But then soon after that, there are these two mutations in these genes MUT-Y. And so this guy increases the mutation rate by a factor of about 150. And these guys both reduce it by another factor of 2 or so. And you can see these things sort of coexist. If you look, they're sort of exactly anti-correlated here. Basically, everyone that has MUT-T has either one of these two, all right? But they're sort of maintained to coexist for like, I don't know, 25,000, 30,000 generations or so. All right, and we can independently estimate the mutation rates. All right, so this is the mutation rate of just the MUT-T, and these guys MUT-Y reduce it, as I said, by about a factor of 2. So these are all kinds of things to start to understand. So I just have a few minutes left, and I want to sort of raise one other kind of point. So in these kind of experiments, we have this sort of incredible power of looking at the time course and looking at replicate populations. So, I mean, in our yeast populations, we routinely look at thousands of these populations over time. But in many natural populations, we don't have that luxury. Instead, all we can do is sequence individuals from a given point in time, typically the present. And what we see when we sequence them is various patterns of genetic variation. And what we want to understand ideally is how does selection and other evolutionary processes shape these patterns of variation so that we can try to start to address the inverse problem. Basically, what we would like to be able to do is look at this kind of data and infer things about how selection has acted in the past, because that might highlight, important events in the history of human populations, having to do with adaptation to agriculture or various interactions with, malaria or the Black Plague or other things like this. So this is a major problem, obviously, to try to do this. And basically, the only thing that is known is if you have a population in which all mutations are exactly neutral, they have no effect on fitness, okay, we can predict what we expect to see in this kind of data. And have any of you heard of this neutral theory of molecular evolution? Okay, there are like probably 100,000 papers or more on this. It's an enormous field, right, going for a long time. Anyway, if you make the assumption that all these things are neutral, okay, then what you can do is you can think about the genealogy of a sample of individuals, sort of ancestral history, and the mutations independently. And so basically what people do, imagine that you sequenced a bunch of individuals. That's these individuals here. We can write down the probability that they have some ancestral history, and then we can imagine mutations occurring randomly on that genealogy, okay? And so this is the whole framework. It's known as coalescent theory for interpreting this kind of sequence variation. We have sort of robust expectations for what this should look like, what any statistic you care to describe from this kind of data would look like under this kind of neutral model. And one can look at things like, how would demographic history, like the migration out of Africa, or very popular nowadays, how would sex with Neanderthals affect this, right? And things like that. So there's quite a lot known about this, but this whole approach basically breaks down whenever there's selection, right? And the reason is obvious, right? You can no longer think of the genealogy and the mutations independently. If there's selection, then the fact that there's a mutation here changes the probability of this genealogy. All right? And so we've been developing ways to try to do that, because what we want to do, so here's an example of an experimental genealogy. We took one of our yeast populations, and others are doing this with the E. coli populations that I just told you about, and we isolated a bunch of clones from generation 1,000 of this experiment, and this is the genealogy, and each of these are the mutations, okay, indicating the genes. And so we would like to be able to look at this kind of genealogy and say, oh, this is a genealogy that's clearly adapting rapidly, right? And so the framework that we've been using to think about this basically is sort of similar in spirit to what I was just telling you about for this forward time problem of the probability a mutation will fix. Basically, the idea is, again, we have all the stuff going on in the population, and we imagine sequencing a few individuals, all right? And what we're going to do is think about sort of the joint ancestry of these individuals, the joint distribution of the ancestry and the fitness of these individuals, okay? Now, the idea is kind of like this. Maybe the easiest way to think about it is this. How many of you guys have kids? This is my standard thing. Okay, how many of your parents have kids? Right? So the idea is your parents are fitter than you are, right? On average, right? And your grandparents are fitter still, right? Because they've their offspring have survived, further into the future. So if we look backwards in time at the ancestors of these individuals, okay, they will have come from a more fit than average subset of the ancestral population. Okay? And we can precisely try to calculate what the probability is that they come from some particular subset of the population. And the further back we go in the past, the smaller and smaller, fitter and fitter subset of the ancestral population the ancestors will be from, right? And at some levels, it's completely unsurprising, right? That's what fitness means, right? The fittest individuals in the past are the guys that are likely to leave offspring into the future. That's what fitness means, okay? So basically what this is going to look like is that as we go back into the past, it looks like a reduction in population size as we go into the past, this action of natural selection, because you're being descended from a sort of fitter and fitter subset of populations in the past. And so this is great, but it's also kind of problematic because it means that you couldn't really tell natural selection from actual changes in population size in the past, which, for instance, in humans certainly happened. We also have other effects which are a little bit more technical, okay, having to do with exchangeability of different branches of the genealogy and so on. The upshot is that these kinds of effects lead to these kinds of skewed branching and these kinds of trees that look like this. And you see them certainly in our experiments. And one of our collaborators, this is a figure from a paper by one of our collaborators on this work, Richard Nair, showing similar sorts of patterns in influenza, which is, of course, rapidly adapting to our immune system. And so what we're trying to do is build a theoretical framework for predicting the shape of these trees, which then can eventually become a method for inference. And I'm almost out of time here, but just to sort of go back now to a question that was raised right at the beginning, of course, all of this sort of thinks about purely asexual genomes, or chunks of the genome that aren't recombining on the relevant timescales. And so one of the sort of active areas that we're thinking about now, and others are thinking about as well, is how we think about realistic genomes in sexual organisms such as ourselves. And the basic intuition is that if there are lots of recombination events on the timescales of common ancestry, basically this is the time to the most recent common ancestor of a sample, if there are lots of recombination events on that timescale, we can think of two sites as independent. So two sites that are distant within the genome, this will be true. And on sites that are sort of close together relative to that, we can think of things as effectively asexual. And so then we sort of want to think of the evolution of genomes of things like humans as this collection of linkage blocks. And these sort of linkage blocks, the size of the linkage block is determined by the process itself. So there's a sort of self-consistency that has to be done here to figure out what this is. But basically, we can think of this sort of evolution as chunks of asexual blocks that are freely recombining amongst themselves and basically evolve independently. And so we've been working on experiments. This is really a slide that's much more appropriate for a very different audience. But we can maintain experimental populations of yeast, both sexually or asexually. And there's a complicated method to do this, but basically you kill yeast if they don't have sex on demand. And yeah, it works pretty well. And I'm out of time, so I'm not going to give you the details of this, but just to sort of show you how big a difference it makes. At the left here are a couple of examples. These are the same kind of figures I showed you before over much shorter time scales. These are a couple of asexual populations, and these are sexual populations. And the basic take-home message of these figures on the right is that sex makes these figures look a lot more messy. There's a lot more sort of stuff going on. And that sort of, in a sense, is exactly the point. It's decoupling, not perfectly, but somewhat decoupling the fates of these different mutations by allowing them to recombine somewhat. So there's still some complicated correlation structure, but it's less than you see in the purely asexual populations. All right. So I'm not going to get into all the details. So I'll stop there. I just want to acknowledge a whole bunch of people from my lab, particularly the sort of work on Rich Lenski's experiments that I was telling you about was carried out by Ben and by Mike McDonald here. Thanks.